All right. Hello. Welcome. This is Rebecca, and I'm going to be going over our Tiny Earth research project that we did this semester. So, what is Tiny Earth? Tiny Earth is a student research experience that involves collecting and testing soil samples for presence of antibiotics. The goal of this is for research purposes, for testing for antibiotics that can be further researched in a lab. So, Tiny Earth is a research collaborative based out of Wisconsin, and they partner with students, professors, and universities across the United States to conduct experimentation in their local area. And this project aims to address the diminishing supply of antibiotics. So, the first step in this project is soil collection. Again, we're testing for antibiotic presence in soil, so it starts as simple as collecting a soil sample. So we took our sample from River Park. It is part of the Chicago Parks District. And we just recorded the weather conditions of the day 4 degrees Celsius, 52% humidity. And then on the left here is a picture of our sample. It's a sterile tube, and we dug about 3 centimeters below the surface to obtain the sample. All right, next is dilution plating. So, how do we get our soil sample onto a plate to observe the bacterial colonies present? We dilute it. So, we mix 1 gram of our soil sample with saline to create a stock sample. We diluted this 3 more times in order to reduce the concentration of the bacteria on each plate, making it countable. So the goal is we want one plate where we're able to count all of the colonies. So as you can see, this first dilution, there's too many colonies to count. By the time you get down to dilution 3 and 4, we can very clearly count the colonies on those plates. So we plated the dilutions, placed them in the incubator for the bacteria to grow, and then this is all of our growth. Next, we want to patch plate. So after we did our dilution plates, we identified the bacterial colonies of interest. So I'll go back just one more. So all of these separate little dots are the bacterial colonies. We picked about 20 of them that interested us, and then we combined them on one plate. This is called a library or a patch plate, and this isolates the colonies so that we can use them for further testing. So this is our patch plate. I said no spreader bacteria present. In the slide before, you can see a little bit of spreader bacteria on these first two dilutions, but this is a good clean plate. So next step is the antibiosis testing itself. We wanted to test to see if any of the bacterial isolates that we patch plated would produce zones of inhibition when plated with some of these bacteria. So we used the escape bacteria, which are 6 main bacterial classes. And what we did is we spread a thin layer of bacteria on the agar that represents one of these 6 bacterias, and then we redid our patch plate on top. So we added all of our colonies in our library or our patch plate on top to see if it would produce zones of inhibition. So here is that. So this first trial we just tested on E. coli, Bacillus, and Staphylococcus right here. So these are our libraries after incubation. You can kind of see like that bacterial lawn, and then you can see our patches on top of it. So between all 3 of these plates, we only had one sample that showed any amount of antibiosis, and that's this one circled right here. That was sample 11 because each of these um, places on the library coordinates with a sample number just for continuity throughout the project. So sample 11 on the bacillus plate had that little zone of inhibition or that clear zone where the bacterial lawn isn't seen anymore. So moving forward in this project, we are only going to use sample 11. It is the only sample that we had that showed this. So moving on, purifying the patches. So as mentioned, our isolate of interest was sample number 11. We used the streak plate method to streak plate sample number 11 to determine how many colony types it has. So as you can see of this picture on the right, isolate 11 had 2 different colony types. We've got this small white colony, and then you can see there's these like bigger, we'll say medium-sized translucent colonies. So after we streak plated it, it allows us to have a closer look at what's really going on in that one like patch sample, and we had 2 different colony types. So now we replay. So me and Deasia are partners, and like I said, only sample number 11 is the sample that we're working with from now on. And it now has 2 new identities because we determined it had 2 different colony types. So this is how we labeled their identities. We have our initials, then we said 11W for the small white colonies and 11T for the translucent colonies. So we replated these colonies separately on a community plate. And this right here is a picture of the community plate and who samples what in what spots. So these are samples from other groups as well. They were each labeled depicting their colonies of interest that had also been purified. So this is the labeled map, and then up here we just made 2 copies of this community plate. So me and Deasia's samples are A and B, so you'll see us right here and right here on each of the plates. That's our 11W and 11T. So now we want to verify it. We saw that it produced that zone of inhibition first, but let's verify it. Will it do it again? So we reconducted the antibiosis testing now with the community plate against 6 different bacterial strains. So in the first testing round, we only tested 3, Bacillus, E. coli, and Staphylococcus. This time we tested all 6. This allows us to confirm the strength of our original results and to look at a bit of a wider bacterial spectrum. Unfortunately, our sample displayed no resistance this trial. It showed no zone of inhibition, even on the Bacillus plate, which is where we originally saw it. So that was the end of the road for our sample, unfortunately. But you do see right here in this picture that a lot of our group mates that we were with on the community plate did display resistance. So all of these that are like dotted in a circle are the samples that displayed resistance. And out of the 6 bacteria we tested, only 4 of the bacteria had these producers. So even though my personal sample didn't show any resistance and it's done, it's still cool we worked with other groups and, finished the project with them, kind of adopted in their samples as our own. So this is a look at the data. So this table shows the samples on the community plate that showed resistance to any of the 6 escape bacteria we tested on. So on the left, you see how we labeled it, the sample ID. It's the person's initials and then their sample number. Their location on the group plate, which is that big map that I showed. And then we have all of the 6 bacterias right here. And then we wrote if there was any resistance and if it was weak or strong. And then each sample from the community plate has its own isolate ID, and that's part of the Tiny Earth database. So we marked the zones of inhibition as weak or strong and totaled the number of target bacteria that each isolate ID inhibited, which is right here. So again, our sample 11 showed no resistance. We're not on this table, but the other samples on the community plate are on this table. And we got to kind of walk alongside them in this part of the experiment. So the conclusion, what did we do? What does it mean? We plated our soil sample and we found many bacterial colonies of interest. That was awesome. We tested the colonies of interest against escape bacteria. Sample 11 showed some antibiosis. We retested, and unfortunately sample 11 did not show that same antibiosis, and it was just deemed a weak and an ineffective bacteria, thus ending its journey. So since our sample didn't go that far, it wasn't really something that's going to get sent into Tiny Earth for future research. It is in the database. What we did in the experimentation and what we found is in the database, but it's not necessarily getting sent to Wisconsin for more professional researchers to look at. So, our sample may not have struck it big. It's in the database alongside 13,000 other samples, and that's that vast resource for antibiotic discovery. So this is a public database that holds the records of soil samples, culture conditions, and inhibitory bacteria to be used for further antibiotic discovery research. And then this is our resources, just the Tiny Earth website. Thank you for listening.